My name is Thor Warnken, and this is my project on photosynthesis. Photosynthesis consists of two different processes that take place in the chloroplast. Each process's products serve as the reactants in the other process. A photon of light strikes one of the pigment molecules in photosystem 2, boosting one of its electrons to a higher energy level. As this electron falls back to its ground state, an electron in a nearby pigment molecule is raised to an excited state. This process continues with the energy being relayed to other pigment molecules until it reaches the P680 pair. It excites an electron in this pair of chlorophylls to a higher energy state. The electron is transferred from the excited P680 to the primary electron acceptor. The resulting form of P680 is missing the negative charge of an electron as P680+. An enzyme catalyzes the splitting of a water molecule into two electrons, two hydrogen ions, and an oxygen atom. The electrons are supplied one by one to the P680+ pair, each electron replacing one transferred to the primary electron acceptor. The H+ are released into the thylakoid space. The oxygen atom immediately combines with an oxygen atom generated by the splitting of another water molecule, forming O2. Each photoexcited electron passes from the primary electron acceptor of photosystem 2 to photosystem 1 via an electron transport chain. Each component of the electron transport chain carries out redox reactions as electrons flow down the electron transport chain, releasing free energy that is used to pump protons into the thylakoid space, contributing to a proton gradient across the thylakoid membrane. Potential energy stored in the proton gradient is used to make ATP in a process called chemiosmosis. Meanwhile, light energy has been transferred via light-harvesting complex pigments to the photosystem 1 reaction center complex, exciting an electron of the P700 pair of chlorophyll A. The photoexcited electron is then transferred to photosystem 1's primary electron acceptor, creating an electron hole in the P700, which we now call P700+. P700+ can now act as an electron acceptor, accepting an electron that reaches the bottom of the electron transport chain from photosystem 2. Photoexcited electrons are passed in a series of redox reactions from the primary electron acceptor of photosystem 1 down a second electron transport chain through the protein ferrodoxin. Enzyme NADP+ reductase catalyzes the transfer of electrons from FD to NADP+. 2 electrons are required for its reduction to NADPH. Electrons in NADPH are at a higher energy level than they are in water, so they are more readily available for the reactions of the Calvin cycle. Water that has been split by photosystem 2 and 6 hydrogen ions are pumped through ATP synthase, which result in one hydrogen ion and ADP plus an inorganic phosphate group turning into ATP, which can go into the Calvin cycle with NADPH produced from NADP plus reductase. First phase of the Calvin cycle, called carbon fixation. The Calvin cycle incorporates each carbon dioxide molecule one at a time by attaching it to a 5-carbon sugar named ribulose biphosphate. The enzyme that catalyzes this first step is ribisco. The product of the reaction is a 6-carbon intermediate that is short-lived because it is so energetically unstable that it immediately splits in half, forming 2 molecules of 3-phosphoglycerate for each carbon dioxide. In phase 2 of the Calvin cycle, reduction, each molecule of 3-phosphoglycerate receives an additional phosphate group from ATP, becoming 1,3-biphosphoglycerate. Next, a pair of electrons donated from NADPH reduces 1,3-biphosphoglycerate, which also loses a phosphate group in the process, becoming glyceraldehyde 3-phosphate, or G3P. The electrons from NADPH reduce a carboxyl group on 1,3-biphosphoglycerate to the aldehyde group of G3P, which stores more potential energy. For every 3 molecules of carbon dioxide that enter the cycle, there are 6 molecules of G3P formed, but only 1 molecule of this 3-carbon sugar can be counted as a net gain of carbohydrate because the rest are required to complete the cycle. The cycle began with 15 carbons worth of carbohydrate in the form of 3 molecules of the 5-carbon sugar rubisco. Now there are 18 carbons worth of carbohydrate in the form of 6 molecules of G3P. 1 molecule exits the cycle to be used by the plant cell, but the other 5 molecules must be recycled to regenerate the 3 molecules of rubisco. The last stage of the Calvin cycle, the regeneration of the carbon dioxide acceptor, a complex series of reactions, the carbon skeletons of 5 carbon molecules of G3P are rearranged by the last steps of the Calvin cycle into 3 molecules of rubisco. To accomplish this, the cycle spends 3 more molecules of ATP. The rubisco is now prepared to receive carbon dioxide again, and the cycle continues.